Good morning. We on, right? No? That microphone is on, but the one in the room is not on. Ah. Hello? Yeah, okay. Welcome, good morning. Today I will talk about comparative modeling. The slides that I'm going to show today are exactly the ones on the web because I copied the wrong file and don't have them here, so I just downloaded it from the web, which is unfortunate because there were some what I then felt were important additions. Okay, now I can't advance. Next week, Tuesday, student assembly, no lecture. Thursday, Ascension Day, no lecture. So the next lecture in this room will be Tuesday in 10 days. Get used to the fact that I don't have my advancer. Let's recap multiple alignments. And let's just begin with a view of proteins 3D, 2D, 1D. Same information here, although it's one dimension less. Reduction of the information that we will talk about in terms of prediction. Similar proteins have similar shapes. As you see here, a family of about 50 proteins that all have a similar function and essentially similar shapes. There are different programs for the alignment of proteins, for the comparison, and the first level of complexity is simply you compare two strings. The way you do that is you essentially try against all possible solutions, brute force dynamic programming, and then you try to find ways to speed that comparison up. Essentially, most of that part is still a simple string comparison. Then we get to the level of biology. The level of biology means essentially you no longer put equal weight to A and G as you put to I and L when you match these two letters or don't match these letters. But you have a comparison matrix that specifically tells you what is more likely to be an exchange residue against another, maintaining protein structure. The next level of biology really is to build up families of conservation, families of positions that are more important to conserve than others. Profiles, they are called, or PSSMs. And now, at that level of complexity, what you take is take the sequence and align it against such a PSSM. I mentioned there are 4 entirely conserved columns here. When you come with a new sequence, you want to match those. Next level of complexity is rather than comparing a sequence against a profile, you essentially compare two profiles computationally. That is the same effort. The problem is writing the algorithm is more complicated because you have too many options. The way you simply can compile the vector product between two columns. That's all it boils down to. And the complication is sitting in this simple fact. When you look at how far we get with the alignment and what can we actually cover in terms of sequence identity, so if two proteins are 100% identical, it's the same protein. Everything is easy to align them. The more sequences diverge, the more difficult the alignment problem gets, and the more the finding of relations get. Finding of relations in the course of this lecture means similar in structure. Up here, everything can essentially be done by sequence-sequence comparisons. Strings plus substitution matrices plus a little bit on top. In this region here somewhere, we get into the region where we need sequence profile comparisons. So we do pairwise comparisons a long time. You build up families, have a specific profile, and then you go out and search again. And further down here, you really need profile comparisons. I've also told you that in terms of population, I'm not used to the pointer, the advanced are not working. In terms of population, most of the proteins of similar structure sit somewhere in the midnight zone where comparisons from sequence alone can actually not really reach. Thanks to Andrea for some of the following slides. Comparative modeling, homology modeling. So the background to that is that proteins that have similar sequences have similar 3D structures. Let me just show you an example, but again, the background to that one really simply is the dogma that I talked about before. DNA makes RNA, makes proteins. Proteins are the machinery of life, and they must have the same structure in order to do the same function, and that essentially is it. In principle, we assume that, no, we don't assume. Many proteins, all they need in order to fold is the chain of amino acids. Typically, this could be 80 residues, 80 amino acids strung up. You throw them into a solvent, and they adopt a unique three-dimensional structure. That can be done in the test tube, and you repeat, and every single time you get the same three-dimensional structure. That implies that all the information about the folding is contained in that sequence and the solvent, right? So that means that in principle I should be able to, maybe not as a magician, but as a physicist, take energy into account and do that in a computer, in principle. Turns out it's not that easy. But let's first look now again at this question of when I look at two different proteins with slightly different sequences, how do they look in structure? Here is one particular protein, the so-called oncogene KRAS. Oncogene, because it was discovered as one of the first proteins that was very relevant in the generation of cancer, in the onset of cancer. You see the standard elements that we talked about before. You see these helices here, 1, 2, 3, 4. You see the beta strands that come together and form a sheet in the middle of it. The helices group around it. And you see a substrate being bound. Now here, yes?
How does the colors?
The colors in this particular case simply help you to guide you through the way the chain runs. So here color is used. There are two ways to answer your question. So the question is, what is the color scheme here? And answer number one, it is supposed to make the figure look nice. Answer number two, actually it follows the chain. From the begin, so the redder, the more to the end of the protein you go. And that sometimes helps you when you go visually through the trace. You simply start here and you go further back and you see that the color changes. But you can immediately appreciate the way this works. That again is important because it has to do with topology. Topology is important because of functional reasons. So following the chain, so where's the beginning of the protein, where's the end of the protein, visually on the three-dimensional structure tells the structural biologist something about that particular protein. That's why they color it that way often. Okay? Now the coloring changes slightly here. We are overlaying a protein in red that in fact is the same protein, but it has one particular point mutant. So at one particular residue position, you have another residue. And that other residue, you had at position number 12 a glycine. So that, as you remember, we start somewhere here. And that glycine now has been changed into a cysteine, G into C. That's the only change. And you see, so it's at the beginning of the protein that you may remember where the blue was. So that's roughly somewhere here. And you see this is essentially the only region where it differs. Where the two structures, so the red again is now the other one, the mutant structure. And you see that in this region here is entirely the same. The binding site is the same. Here, this binding site at least. Here you have some effect. So on this, the strand is a little bit twisted or a little bit turned, simply because the interaction is a little bit different, because there's a different residue somewhere here. Okay? That's the only change you see. Now we get to the next level, and now two colors, we lose colors, now we have only two colors left, and these two colors really compare two different proteins. One is KRAS, the human KRAS, and the other one is called RASH. They have 85% of their residues identical. Okay? Of 85%, so most of what I showed you, this is in the realm of most of these simple toy examples of sequence comparisons that I showed you. 85% is a high level of similarity. It's something where we typically sure that we can do the alignment and everything is sort of trivial pairwise alignments. On the level of the structure, you see that these helices move slightly here, but essentially this region now still is the same. The binding site is still very similar. Okay? Two different structures that are 85% sequence identical to each other. We go down in terms of sequence identity, and we go from human to fly. So now we find the protein RAB6, which is the closest to KRAS, to the human KRAS in the fly, in Drosophila. And so this is a big jump here. So now it's only 30% of the residues that are identical. And still, I'm now confused about the coloring. I believe before we started with the two green ones, and the RAB6 now is the orange structure in this here. And you see that still, essentially at 30% sequence identity, still those three structures look very, very similar. And as before, most of the similarities are here, most of the changes are here. So that seems to be a trend that in this particular case went further. Let's go down, back to the situation here. So we are still in a realm where we have sequence to profile comparisons that do the job. What if we went further down? So further down, purple now, we get to 19% sequence identity, we get to the bacterium. So now we leave the realm of eukaryotes and go to a protein that no longer actually has the same function, that now does something very different. But even the purple one, you may feel still, okay, now I show you an image that is beginning to be very confusing. I have 4 proteins overlaid here. But even the purple one still fits very well to that structure. Mind you, protein structures have an immense variety. It doesn't have to be the same number of helices and strands. They don't have to be packed the same way. And essentially all of that is the case here, even at 19% sequence identity. So sequence identity implies structural similarity. Here back to some image from Christina Rengo of the diversity of what you could achieve with a sort of similar number of residues. So what I showed you there, the similarity is absolutely not trivial. This is just some subset of the protein structures, and most of them look very, very different from that similarity. Now, this observation so far, similar sequences lead to similar structures. How could I use that to predict structures? Let's call it a hack prediction. What could I do? So I would argue that essentially you have all the tools I have presented to you in the lectures that I've given so far, all the tools that you need in order to come up with the first method that sort of does a hack on predicting structure. How can you do that? And what I'd like you to do again is form groups. Today it looks a little bit easier, except for some exceptions. Just sit together with your neighbors or neighbor and chat for two minutes or something like that, and try to come to an answer to the question: how can we use what I presented to you so far in the lectures to predict structure in a hack way, not energy? Something that has to do with sequence comparisons, and something that uses the fact that I just showed that similar proteins have similar structures. Carlo, could you switch off the camera in the meantime? The solution, but it may not be apparent to everybody else. He says, we can take some chemical. Let me just repeat your words, as far as I remember. I didn't catch them all, and I'm bad at, my memory is not working very, very well. Let's just take some chemicals. And I assume what you mean is proteins. So let's take some proteins for which we know the three-dimensional structure, and then find the similarity, and then we are done. That's what you said, right? So everybody understands that that is the solution? It's not that trivial to understand what you say. You're right. But can somebody translate it such that maybe it's immediately making the ah effect to everybody? Okay. That was an R effect. What was that? Not my computer, that's good. But the projector is on. Oh. Too many question marks for the projector. Another group. He gave the solution, so you should everybody know now. It was not that clear, yes?
We can make proteins such that we know.
Yes?
We can make equivalent sequence that we don't know the structure.
Yes?
And if the sequence is similar, we can more or less predict that the structure will be the same.
Everybody got that. So the people in the vicinity heard it, and it is the same idea. Did you understand that it was the same idea? Yeah, okay. So everybody got that was the, sorry, then I misunderstood how much you can abstract from ideas. So I believe, I take it, everybody got it. So you have a query, that's a protein sequence. You have the database of protein structures, PDB, protein database, that in fact contains proteins for which I know the three-dimensional object, the three-dimensional structure. Each one of them comes with a sequence. Here is one particular one that I find by pairwise or sequence profile comparisons. I establish by those comparisons that these two, the query and the protein of known three-dimensional structure, have similar sequences. From the fact that they have similar sequences, I deduce that they have similar structures. And that's exactly what these two groups said. Now, just one more detail. Implicitly you said that, but just to make it more explicit. So the one in the PDB, this one here, looks like that. So we look up the three-dimensional coordinates of this one. And for that one up there, through the alignment, the alignment, I'm sorry, implies a corresponding position. That means for every residue in this one here, I can look up where that position is in three-dimensional space in the other one. And I just read off the coordinates. Okay? That's in principle what we do. The reach of comparative modeling, so the degree to which it can model or be applied to the database, I'm going to show you in some outdated views, but essentially I believe the ballpark here, the order of magnitude is still correct. What I'm going to show you is a view where the whole pi will signify all known proteins. At this point about 55 million. Estimate is from 99. The numbers from last year about sequences, structures, and so forth are there. So 55 million sequences, about 100,000 proteins with known structure in the PDB. For 13%, so this somehow, the estimate is slightly different because this estimate, 100% is not all known sequences here for 55 million, but 100% on this one is 400,000. So essentially for 13% of all the ones in that smaller set, I have an experimental structure. For 22%, I can do fairly straightforward comparative modeling, which is exactly that technology that you just invented, right? Using relatively simple pairwise and sequencer profile alignments. Here, in this realm, I have to sort of expand into profile alignments. So here the problem gets more to find that this protein of known structure is actually similar to mine. But you see this sort of covers half the pie. Here is the same view where we look instead of at all sequences in the universe, we look at all proteins in one particular organism. And we sample this here by about 20 prokaryotes by 6 eukaryotes. And overall you see that roughly, and again it's not completely today, by some criterion of the comparative modeling accuracy, we get 30 to 40% of all the proteins entirely in sequenced organisms to model with comparative modeling. Let's get back a little bit to concepts now, and to the language that is used. So first of all, there are two different terms running around. comparative modeling, and homology modeling. Essentially means the same thing. The reason why homology modeling may hit you in Wikipedia pages and in old papers is simply that originally the idea was there was an evolutionary relation that I dig out. And that evolutionary information helps me to infer structure. But at the level at which comparative modeling is applied, mostly there is no evolutionary relation. It's just the sequences are so similar that if you randomly create them, most likely they would have a similar structure. So evolution plays a role. Evolution is important, but it's not really the most important for this technology. That's why people came up with this idea of or the word of comparative modeling. More modern, let's put it like that. There's this language of a target and a template. The target essentially is your query, the protein that you don't know the structure for, and the template is the one that you use for which you know the structure. So you use that as a template to model your structure. In fact, again, the database to identify the template from is the protein database. So the database of all proteins that have similar structures, known structures. And we identify it by aligning the target against the template. Then we build a model, and then we assess the model. Now, build the model is what I just said. I, in fact, take every corresponding position implied by the alignment, and I read off the three-dimensional coordinates. Now I have a model. I have a three-dimensional object for my query protein, for my target. Read off the template. But how can I assess this model? What could I do? And this is not something that has been answered directly in previous slides that I presented. So what could you think about that could help you to see how good that model actually is? Could you think about, throw some ideas at me, how you could check, what would you see that immediately tells you there's something wrong without necessarily knowing how to correct it? But what would be simple things that you could see that are wrong? Yeah?
We expect some behaviors of every protein.
Yes.
Right?
Such as, for instance, proteins, I will get back to that over the course of my lecture. Proteins essentially look like spheres. And if it doesn't look somehow like a sphere, something is wrong. That is one statement, yes. But even much simpler than that.
There are chemical activities that every protein is.
So every protein has to have a function. Now let's go back. You're right about what you say. I'm sorry that I interrupt you. But ultimately what you want to say is, and I'm only repeating it not because you cannot say it, but because I have a microphone. And the camera hears me better than you, due to the microphone. And the point that you make is essentially proteins have a particular function, and what we have to see here is that this particular function is done. Now the problem with that concept, you are absolutely right. The problem with that concept, on that level on which I operate here, imagine so far, for the first part of our lecture, now I get it. It's the PDF. So that's the problem. Okay, I have to have my fingers on it more often. So far, you in the room essentially could have done most of what I proposed to you if I had given you the simple tools. In principle, you came up with a solution. Now, understanding what function means is highly complex. And saying that this is right or wrong means that you know for every single molecule what it does, and that we don't. For half of the human proteins, we do not know function for half of the human proteins, for way less than that. We know structure by comparative modeling for more human proteins than we can say something about function about. And that already tells you that this idea may be a right one, but we need something first. Maybe tomorrow. Or, in fact, to stay here, not tomorrow, but next semester, I will talk about that. But not over the course of this lecture, so this is beyond us. Okay, some simple things. Again, the question is, you build the three-dimensional model, so you simply copy the coordinates, and now you ask yourself, is it right or not? What are simple things you could see that are totally wrong? If the overall shape looks very different, is one solution. Any other things? Simpler. So you're trying to do the shape of a protein. Could you think about things that could go wrong in that model? Maybe I go back again to some of these superpositions of Although this is a little bit misleading, but maybe the other one is even is better even, because it's more, those things are a little bit less similar. Yeah, but maybe you don't see that. They're less similar. But any other idea what you could immediately check? So if I simply copy the coordinates from that and use it to model that, what could immediately go wrong that you could immediately see whether the model is right or not, or something in the model can point to your position in the model that are not right and could help you to, in fact, assess the model. The direction. So if you, direction is a complicated story. That's a very complicated story because, so let's go back. The sequence alignment implies a direction. The sequence alignment implies which residue you correspondingly look up. So that means from the alignment is implied a direction, from the direction is implied a lookup in the model, from the lookup in the model is implied a direction, and you close the circle. So you will not get into trouble with the direction. Because at every single step you implicitly watch for that. That will not go wrong. The topology could. That is more complicated. Topology has to do with how loops are connected. That could go wrong, but much simpler. Well, remember in the sequence alignment comparison, we have insertions and deletions. We have holes. Proteins don't have holes, right? You're right, they have a nice shape. But one aspect of this nice shape is they don't have holes in them, or rarely. Because why would they not have holes? What's the problem with a hole in a protein? It breaks the structure. So the hole itself does not quite break the structure, but it opens the door to a break of the structure, because solvent could come in. This is in a solution, this entire thing. The moment you have a porous hole, water gets in. There's an entropy issue. There are issues that will essentially break up this protein. So in fact, very often you sort of cut by a scissor or by a mutation to the core of the protein is the inside, and the surface is clear. It's the part that is accessible to solvent. And if you break a few crucial bonds at the core of a protein, typically you destabilize the protein. Not because you really break the structure in that sense, but because the solvent comes in. Okay? So that would be things you could immediately check. Are all atoms connected? Are there holes created? Are there clashes of atoms? Does the model imply in one particular position that, for instance, this helix here is implied in the model to be here? And then between the two parts of the helix, this one here and the green that then moves here, there's so much space. That is a hole, that is something that you could not bridge. That would be clearly a mistake or a problem. Or you place a bunch of positively charged residues next to each other. Impossible. They completely repel each other. So those are the kinds of things, simple energy functions, simple criteria about how far you can bridge with a single amino acid without breaking the chain, without introducing holes, are things that you can use to assess the model. Once you have assessed the model, the problem gets more complicated. So you may say my model is good or not good. If you said it's not good, how do you change it? Well, in this particular case here that I assumed is not shown on this image, I just take the image to bring that idea, assume this would be the green, and you would know that you would have to bring the two greens more together. That will do something else in the model, and that may not be possible. But this is something that you may want to try. And then, once you tried that, sort of move the coordinates essentially around in the computer, and then try to see what the energy functions of that one looked like. In terms of the way comparative modeling performs, at some point here, up here, the speed of modeling is the crucial thing. So if two proteins are very similar to each other, you really want to model all the detail. Model all the detail means you look up the coordinates, that part is easy. But then you want to assess the model, and you want to refine the model, and that is the part that becomes, because then you do energy calculations, and that becomes the problem here. Somewhere in this region here, the quality of the model becomes the problem. The quality of the model means that the two structures actually are not longer the same. There is some movement. I showed you that between the mouse, no, it was a fly protein and the human protein. They were still fairly similar, but sometimes there can be more movement. But even between human and the fly protein, even there was some difference. And even there, the difference between these structures means that your model cannot get better. Your model can, if you use the fly protein to model the human protein, you will never get better in your model than actually to really get the fly protein, which still is not the human protein, right? So that is the quality of the model here. One level down, further distance in terms of sequence identity, the main problem becomes the alignment. So essentially, now your programs that align strings make mistakes. And they make so many mistakes that the corresponding residue, so I think that these two residues correspond to each other because the alignment overlaps them, is wrong. And the more wrong that goes, the more wrong goes the lookup of the structure. And that you cannot correct by the things that I told you before, by assessing or refining the model. Because refining or assessing the model always assumes that is still the right position, you just need to move this position around. Switching to a completely different position opens up the can of complexity. And that is the problem that you get at this level. When you go further down here, you get into the twilight midnight zone, and there the problem really becomes to detect the similarity. Okay, let's just look at a couple of state-of-the-art methods that do these things and give you some examples how they do that. The first comes from the Charlie lab at UCSF. It's called Modeler. And we begin, we find, this essentially means I find a protein of known structure that in terms of sequence looks similar. I refine the alignment here, the target and template alignment. I build a model, I do the evaluation. And again here, as before, I say the model is right or not. If it is good, fine. If no, now they go into different ways here. So some point in here could be what I told you, do some energy calculations, and we get back into, they do that in an earlier step. Instead, at this point, when the model doesn't look fine, what they say is redo the alignment. So maybe there's an alternative way in which you could have compared those two strings. Yes, I still believe, in fact, that this is the structure, but let's just move things around in the alignment and then see whether it possibly gets better. The alternative is, with growing databases of proteins of known structure, you have an opportunity that maybe there's another one in there. Remember I showed you RAS, RAP, and the fly protein. So typically you would take, if you want to model KRAS, you would take RAP, because that is 85% sequence identical. It's very similar. But maybe in fact the fly protein at 29% sequence identity, for some aspects of the RAP structure, would be the better one. So maybe you try a different template, which still assumes it's the same structure and so forth. It's just a different one, a slightly different one. And maybe that would work better, because there's something unique to your protein that is modeled better in that one. Okay. The one issue here is constraint satisfaction. Constraint satisfaction, ultimately, it has to do with local spatial constraints. And local spatial constraints are exactly what I said before. How far apart neighboring residues are, how much contacts you have, how much open spaces you have in some environment. Are you bending things too much? Satisfies spatial restraints ultimately also has to do with the issue that you brought up, which is the overall shape seen from the outside. So the whole thing at the end should look round, and all of that is considered here. And in this consideration, the model is changed at every single point slightly. Here's an example for a very, very simple aspect of it, which is simply the bonds, the differences between two cores, or C alphas as they are called, course of a residue to each other of two different. So we're looking at two residues, I and J, that are close in space. And this here is the distribution that we observe in the database of known pairs. And we simply, when we get into our model, we simply try to fit that model to that distribution. And the deviation from that observed distribution gives you a sense of wrong model, right? This is the way you go from sort of bar observations to some overall model that fits the black into the bars, the observed bars, and uses that black fit to then compile a pseudo-energy-like potential that allows you to assess the model. Procedure is clear? Yeah. So then I said possibly you could use a different model, so a different template. You could use another protein for which the structure is known that might also help you. And then you in fact could look at this in terms of the best fit, not in context of the sequence alignment. So not I start from KRAS in human, I find another KRAP in human that is 85% sequence identity, hence I take that as the first model. But going to the alignment of, for instance, the fly protein, 30% sequence identity, and say, well, for the fly protein, all of these constraints look better. So I take it just because it looks more like a three-dimensional object that I expect, not because from the sequence I assume that this is the closest. This is the idea here. There is a lot of methodology underneath. There's molecular dynamics, so Langevin dynamics, self-guided MD and LD, rigid body movement, rigid molecular dynamics, rigid minimization. All of this goes on under the hood. There's a lot of physics, a lot of computational biology underneath, 25 years of work, many details right. You do optimizations, high computer need, and then you essentially optimize through it. At some point you get a model that looks okay. You see in the model whether you can accept it, you try to change it, essentially the protocols that I showed. And then there are various details in which you get to the final solution. Here's an example of, that is the error. RMSD is the root mean square deviation. So for every single residue, for every single point, you look at the difference between the structure that you predict by comparative modeling and the one that is actually known. So you pretend that the actually known one you don't know. And this is somehow the score, the energy score, suggesting that your model looks right. And the good news is here that the lowest score typically is also the lowest RMSD. So if you just go dash in your model for the lowest solution here, then typically you find good solutions. In this particular case, you see it's not the best one. So the lowest RMSD would actually be one that, according to your assessment of how good the model is, would be worse, higher score here than this one. But in terms of the real prediction accuracy is worse. So your model cannot always pick, is what this one says here. You make mistakes, but overall the best solution is one, the best solution that your model picks is always the lowest DOP score here, the lowest energy solution, and that is always a very good one, okay? What if the loop is missing? So what if the difference between two structures, here's an example where we have a long loop, here's an example where we have a short loop. What if part of that is completely missing? How can you model that? Let's look at two similar loops. In this particular case, I'm not entirely sure how much they differ. In blue and red, but they're fairly similar. Can we, in fact, predict the right one by just doing molecular dynamics? Let me go back. I said that proteins put into solvent adopt a unique three-dimensional structure, and that in principle I should be able to predict that in a computer. Today we can't, because of the complexity of the issue. We have too many residues. What if we had a loop, so something that connects regular secondary structure, and what if this loop were only 5 residues long? So you have the constraints of the end points of this loop, and in between you have a connection, and one has 8 and one has 9 or 10. Can you get the loop right? And the answer continues to be, most of the time we get the answer wrong. The best methods out there cannot solve this problem. This means that when there's a loop that is completely new, we can absolutely not solve it. Typically the way we cannot solve it by doing molecular dynamics, by doing energy calculations. Since we cannot do it by that, how else could we hope to do it? So I gave you the task, I gave you these 2 points here, and give me 8 residues that find a path in between these 2. How would you do that? And again, think about a simple hack, not assuming that you're the first person in the world, but that you have all the resources to look up something. And it's one of those few cases where Google is not the answer to the question. So you cannot Google the answer in this particular case. But what would you do? Yeah?
Find the most probable.
Probable. How would you find the most probable one? Energetically?
From the data we already have.
That's exactly the idea I was hunting for. So you have a database. From the database you just look up every loop that has 8 residues and see whether that is somehow in a corresponding position, and then you take it. Now, most probable in that once you have one of those, maybe once you have 5 of those, you take the one that has the best energy for the entire environment. Energy means that now you look at constraints or at distances or at connections that you imply by putting the loop into this to the rest of the structure, to the rest of your model. And that's exactly what the most successful approach currently does. Okay, this just repeats what I said before. So most of the time we get some idea about the loop, but mostly we get essentially wrong. Swiss model. So a modeler has lots of bells and whistles, it's downloadable, it's very accurate, and is the tool, state-of-the-art tool for comparative modeling. Swiss model. Comes today from the Schwede Lab in Basel. Here's the website. The underlying philosophy here is it's fully automated. It should work for non-experts. So it should work for somebody who has never used comparative modeling and has never really seen much more of protein structures than you. Hence, it is for people such as you, except for typically this is for biologists, but most biologists have no idea about protein structures. Most biologists would not be an expert in this. So in some sense, you would align very well with them, except for you know how to use computers. And in some sense, the underlying philosophy of it is, the less you do, the fewer mistakes you make. So let's just copy the coordinates as raw as they are, and let's get rid of the bells and whistles. So essentially, the idea is do the alignment, copy the coordinates, and go home. And things got a little bit more complicated from there. In fact, that was the first program of Swiss model that was made available, and that was competitive with many of the existing programs a decade ago. Not only competitive, actually, it outperformed most of the clever competitors. And I believe the cleverness really was in not making mistakes, by making it as simple as possible. Today, this is a small flow chart. Today things are way more complicated, and I will zoom through it because essentially Swiss model adopts a similar philosophy or a similar mechanism as does modeler, with the difference that in the detail some steps are done differently. And this level of detail, I believe, is beyond the level of this course. One example for a detail here is that the alignment is done differently. So one particular point of Swiss model is that it now uses the HH Blitz, HH Search, so the hidden Markov alignments. Yeah?
So we are comparing only proteins for which we have a structure.
No. So in my lecture, this goes, unfortunately, a little bit cross sometimes. When I give you assessments of how methods work, then those assessments are always done on proteins for which I know the structure. I pretend I don't. Comparative modeling works on proteins for which I do not have the structure, but that I can map, means sequence alignment, find similarity to another protein for which I do have the structure. But that is the 30% or 40% that I showed you before for entirely sequenced organisms. For roughly 30 or 40 or 50%, depends a little bit on your cutoff, of all the human proteins, I can find something in the database of known structures that has such a similar sequence, that I believe it has a similar structure, that I believe my comparative modeling would work. Not only can I do that, and not only, as in this example of Swiss model, is it relatively simple, but today, this is by far the best method you can have to predict structures. There's nothing out there, so I will later talk about Rosetta, simply because you brought that up too, about tools that can predict protein structure from sequence alone, molecular dynamics software and the like. So these methods that try to understand everything about the way proteins form, shape, and simulate the folding, and none of them comes anywhere near comparative modeling. Rosetta, for instance, does, because Rosetta now has a built-in comparative modeling package, just like the other ones. And that is why it completely is as good as Modeler, most likely, on the same turf, because it uses the same ideas. But deep down, when you ask what's the mechanism by which all the good, if you looked at all the human proteins for which over the last 2 years structures have been determined, and that until 2 years ago had only been predicted. So if you look at the distribution of accuracy, then almost everything that has an acceptable accuracy comes from comparative modeling. And the major difference really lies in, and that is one difference here to Modeler, in the way you identify the similarities. HH search is a hidden Markov model search that does a very, very good job to get this done. Part of the rest is to refine in using multiple models. That is where Modeler, by the way, is the best. The template selection, again, that is what I said before. So more and more these days, the database of known structures grows, and you have situations where you do not only find one, but several. And then the issue becomes which one to choose. I showed you the historical solution of Modeler was one that was biased more by energy functions. The one from Swiss Model is more biased by functional constraints, and in some sense that's exactly what you said, but slightly different. So looking at what else for my query, for my protein of unknown structure, do I know anything about what it binds? And if some of my, say it binds calcium or it binds DNA, and for one of my templates I know they bind DNA, and for another one I don't, and I take the one that binds DNA. So now this solution here is something that has nothing to do with the physics. It's not an algorithm. It's a solution, a cut on a different level. It also uses secondary structure and solvent accessibility predictions, and that is in fact what the next lecture will get into more detail into, how we can predict secondary structure. Essentially, all comparative modeling methods do that, but Swiss Model does it a little bit more, focusing on a core. So reducing the part of the protein for which you believe that really is the heart of it, and modeling everything around it. So not just as initially take the 3D coordinates, but the next step would be, in my 3D coordinates, what's the most important part of it? What's the core? What's the densest? In 3D, I'm looking at something, I look at this, and then maybe in 3D I see that the densest part is here. So I begin to model first here, and see what that implies for the model. That's another idea here. Okay, after many steps you get a model that looks like that. What I show here is just this set of backbone angles. So for every pair of residues it shows whether that falls into something that we know. And that is a typical model, in this particular case, that looks fairly right. There are regions where this is an energy function over the course of a protein, the residue number, from 0 to 350. And you see that in fact, in this particular case, It's a preference for being in a solvent. You see there is some variation, there's no flat function, and essentially you can see that as a score for error, but all models contain errors. And the challenge for the user of the model is to find the right region. Swiss model, although it has changed over the years, still clearly is automated. It's increasingly comprehensive and flexible, and that always implies increasingly complicated. But still is sort of the simplest way out there. What else could you do? I'm just briefly going to mention a few words. H.H. Pratt. H.H. Pratt comes from Johannes Söding, originally from Munich, down Göttingen, who most of the contribution was really in doing the alignment better than everybody else. And using the simplest comparative modeling program, the simplest being Swiss model, you just look up the coordinates, and not much more than that, but doing the alignment right. Now, the way he did the alignment right, he also used some aspects of secondary structure. So somehow, this is a different approach. You pack, where I said modeler essentially takes an alignment as a first step, and from there on, it tries to operate in 3D. With energy concerns, with whatever is right in shape, whatever is clashes in energy. While this approach here goes much more on the level of sequence, saying, I built into finding my first template already as much as I can about that particular structure. That's the deviating philosophy, essentially. 3D Jigsaw. Now that idea comes essentially from this image here, where you say, in contrast, I explained to you that Swiss model has this focus on a core region and model everything around a core region. Because that is the way proteins are constructed. That's the way proteins fold. 3D Jigsaw imagines that what we have is not really these condensed regions. What we have is just a jigsaw puzzle. We get pieces. Just like in this example of the loop that I asked you, how would you model if I had two constraints in the loop of 8 residues in between? You would look up in the database other loops that most likely would fit, is what you said correctly. And 3D Jigsaw has the same idea. Let's just completely chop it up, the entire database of all known protein structures, into little fragments, and then put them back together for that particular model that I can build. Put models, so in this particular case, you would in fact take your first look up 3D structure by comparative modeling, but then refine by fitting it to fragments. So at every single position, you would begin with a lookup of a 3D coordinate. But then you would sort of refine by picking pieces from this entire jigsaw puzzle. Pieces that fit together. 3D Jigsaw. Assemble the fragments to the 3D structure. What if is peculiar, it comes from Gert Friend, well, it's peculiar in many ways. One way in which it's peculiar, this is written in Fortran. Because it's a piece of software that essentially is written over more than 25 years ago and still runs and gets increasingly complicated. And what if can do almost anything. So this full is one of those old-fashioned programs that allows you to play chess with it too. It's just, imagine 1 million lines of Fortran code. That essentially is something that it is. It grew over the years. What if the underlying philosophy is the following? Assume you have this structural alignment that I showed you before, where, remember I told you that if the green helix would be a little bit too extended, then you would know immediately there cannot be holes. There cannot be pushes. Or if you wanted to ask yourself, what would be the impact if I move the positive charge residue that in fact I know has some damaging reactions for some binding. I want to get rid of that positive residue. What would happen if I did that? And in particular, what would happen if that interaction would be somewhere in the core? That would possibly create a hole. That hole would possibly create some damage. So the entire philosophy of what if is bring the user in a position where they can ask a question like that. What if I cleaved a sequence in silico on the computer screen? What would the impact of that be? So what if then focuses essentially on the follow-up computation. Given a model, given a solution that you believe is right, how would you change it? if you believe that this is wrong, or if you believe that if you wanted to change something to design a new function into that protein, a new binding site, that's the underlying focus of what if. What if was boosted, and this is another statement that I want to repeat, was a similar story for Clustal. I would argue that the value of Clustal W was so high because it had a very good way of presenting its results. For what if, that is Yazara. Yazara is just, you see here, journal covers. So when your program, when your method, your publication, when there's a figure in there that will be printed on a journal, that is one way in which what you do is visible. Everybody is going to have a printed journal, or you're going to see this printed journal. So that's a major thing. And Yazara is just one of those programs that has been used a lot for figures that became famous. I just show 3 here. And ultimately this is, back to your question, what is the meaning of color? Ultimately, because it's nice. This is not necessarily meaningful, but it implies an understanding. And in some cases, this meaning may lead to some other understanding that may be real. It may just be beauty. Clearly, it looks interesting. Clearly, having the opportunity to sort of look at these two, this, again, we're looking at a language that presents a 3D structure here. This one here, this rendering, has nothing to do with the protein structure. But it immediately implies some interpretation in the eye of the one who looks at it. And sometimes when you write visualization software, when you write images, find ways to hook into what you want the face to associate with it. Our ability to associate is remarkable. Just cater to it, learn from it, do something for it. That is what if Yazara. ITASA. The ITASA workflow, that's another one, more modern program. Its main advantage is in combining essentially everything. So combining the energy-based modeling. I mentioned the program Rosetta. That is the program that many people use to essentially model 3D structures from energy principles. It has to do with taking from the database what you have and look at the most probable solution. It realizes 3D Jigsaw, it realizes Swiss model, it realizes Modeler, and somehow is a huge package on top of that. ITASER is essentially the same thing written by a different guy. A smaller community, it's a one-person community. Originally, it was a postdoc who did it. It went bigger from there because it was very successful. And it does many things right that by introducing more modern things. Sometimes you can do better by just throwing away what others have done before, because over the time big programs that grew just aggregated some nonsense. Sometimes it's difficult to do that. Here in this particular case it worked. In most cases where people tried to compete against Rosetta, it failed. No, don't get used to that. This image here, some of you may understand enough French or of images to read that this says this is not a pipe. The image comes from René Magritte, shown here. It's not a pipe. It's a model for a pipe. Well, Magritte has his meaning. I completely misuse this image from Magritte for a different purpose. And that misuse is essentially about a model. The model of a protein is just like this. It's a pipe. But it's not a pipe, okay? It's a model for the pipe. Magritte meant it as a sort of provocative statement. It's not a pipe because the pipe is the object that you have in your mouth. This is an image of a pipe. This is a model of a pipe. This is some abstraction of a pipe. Whatever you call it, it's not a pipe. The pipe is that three-dimensional object that you can touch. So the moment you have an image of something that may be a truthful image, everybody in the room immediately would recognize that this is a pipe, or most likely. Maybe most of you, if I showed you the telephone that I had when I was your age, most likely most of you would not recognize this as a telephone. So sometimes think, oh well, maybe you would. Have you ever dialed? Rotary phones, have you seen outside of museum rotary phones?
Yes.
Where? Okay. Sorry, now I'm this should go off the camera. Is that true for everybody? Yeah? Yeah? You have had home rotary phones? But wait a minute, I thought they died out 20 years ago. You guys are not much more than 20, are you? 22, 23, come on. From my perspective, there's no difference. I mean, is there anybody in the room who is more than 30? No. Okay, I got you there. Is there one who's 25? Oh, 2, 3, oh, okay, 4. Big number. But 25 years ago, there was no rotary phone anymore. Okay. This is a pipe. Back to my main message. I was obviously wrong on the pipe. Do you know what this is? I mean, this LP, the long play disc, so where you scratch and then you have a strange sound and then you believe that is music. And you know that from the disc? Yeah, sure, from the DJs today. This is the rappers. Okay, that one you know. Anyway, so. VHS tapes. Guys, when I interviewed at Columbia, that was 15 years ago. 10 years ago, in the interview for the first time, when we interviewed Columbia new students, Columbia University, they give us always the pep talk. Remember that these students were born at a time when this and that didn't exist. So put yourself in the mind frame of these students. And more than 10 years ago, they said that students now would not know what was there before MP3 players. How come you guys know all of that? You go more to museums or what? Or is that the difference between Germany and the US, or?
Exactly. Yeah.
That's the difference between Germany and the US. Germany's more than 10 years backwards. Okay. Oh no, you're not starting today. Well, but you are, yeah, that's the difference. Okay, I get it. They're younger, the students are younger. Back to that image here. The important point that I want to transmit. We get a model, a comparative model of a protein structure. And the most important first question, I did the modeling, I asked in the modeling, where are things wrong? Once you have the model, you have to turn that upside down. Now you have to ask yourself, it's a model of reality, this is not going to be reality. But the most important question is now, where is it right? What in that model do I believe the most? What's the implication of that image? What does it transmit in terms of understanding the problem? What does it imply for function? What do I learn from the model? And what of that learning, when I go back to that model, do I believe, that I learned from something that I'm really sure about? And what of that learning did I learn from something that could be different? Okay, back to the pipe here. In some sense, not all the elements of the pipe are necessarily the way they are. It must be wrong somewhere. It must be different somewhere. There are some regions that are wrong. If everything would be right, it wouldn't be a model. Where did it wrong? Where is it right? Find the difference. A model, Thorsten says, is something that helps you to interpret biochemical data. A model is something that is a vehicle to get somewhere, not an own meaning or not an own sense. The pipe is what you smoke. The image of this pipe is something that you show others to. They will not understand how it's. How many of you know how it is to smoke a pipe? Okay, this is not the kind of question that I should ask now. But from showing you a pipe, most likely there's some people in the room who have smoked the pipe. And it has become obvious to you when you smoke a pipe, particularly when you're young and do it the first time, that there are certain things going on in your body after the first smoking that you had not quite anticipated before. For me, this was sort of a panic reaction that had to do with all kinds of stuff that came out of me ultimately afterwards. But the image is not implying that. You cannot get the feeling from just looking at the image. Still, image teaches you something. And that essentially is the point I want to leave today's lecture with. Whatever you come up with, remember I had this reality in images. We create with models, we create images. And these images are as long good as we know what of the image supports what I have in my head. So I have to sort of be very self-reflective and remember that's wrong in the image, that's right in the image, that's why I come to that conclusion, that's what I learned from the image. And go back, is that actually right? That's the scientific procedure. Thank you for your attention. Have a good weekend.